So first up is primary. And the main difference between these two is primary you sort of inherit. You're born with it. It's congenital. You don't pick it up. It's not infectious. Secondary doesn't always have to be infectious, but secondary, it means it's acquired. You come across it, or you're exposed to it later in life. Okay? Now, that later in life could be after the genes have been determined. It could be only that later. Does that make sense? Meaning that if congenital, it's not a congenital defect at the level of the genome, and the developing fetus doesn't have a primary congenital defect at the level of the DNA. And I'll explain this here in a minute. But because of mom or dad, but mom is exposing the fetus to some sort of deficiency disorder, great example would be HIV. Okay? Then the developing fetus could have an acquired secondary immunodeficiency disorder almost instantaneously. But the difference here is congenital means it comes from the genome, and acquired means that you pick it up somewhere else. It's not from the genome. It wasn't coded that way. Okay? That's a good distinction between the two. So if you look at primaries, and these are really busy slides, but they're actually super simple. Okay, so we're going to walk through these. This is an overview, and this is from your textbook, and it looks super, super busy, but we're going to pare it out very, very specifically. These primary immunodeficiencies are single gene defects. So they're defects that happen at the level of the genome, and they lead to challenges or complications. Now, the single gene defects that we're going to look at are going to be B cells, T cells, and both. We're not going to have time, we're not going to cover complement and phagocytes, but those kind of defects do exist. So there are patients that do not make B cells. There are patients that do not manufacture T cells. There are patients that do not manufacture either. Right? And there was one a long time ago that was popularized in the media. It's called Bubble Boy. Okay? And so, again, with primary, you are more dangerous to them than they are to you. Does that make sense? You're not going to catch, right, from a primary immunodeficiency. There's nothing infectious about that. You are extremely infectious to them because you are carrying all sorts of cool and crazy, interesting diseases around, right? So if you look at this, you can start to see these little red squiggly lines that show where these known defects happen. These are like single gene mutations that take place. So if we blow up the first one in higher magnification view, so I'll back up here for a second. We're actually going to be right here, okay? So that's this right here. You see that? So the first one I want to talk about is X-linked agammaglobulinemia, Bruton disease. Okay, this one's actually found here at the very bottom of the slide. And the symptoms are a low or an absent production of B cells and plasma cells. And the reason is because you've got a pro-B cell that goes to a pre-B cell, and if you back up, this pre-B cell right here is supposed to become an immature B cell that gives rise to mature B cells that manufacture M, G, A, and E, immunoglobulin. So if you have a mutation here, and this is at the level of the bone marrow, so this little box is the bone marrow, and your mutation, I'll blow it up in higher view, is right here, X-linked Bruton disease. You don't make really any B cells, but you have a completely normal T cell response. Does that make sense? So in this particular example, with our B cell example, right, we have a problem where the heavy chain is synthesized, but the light chain is not. So we don't actually get functional immunoglobulins. We get normal T cell response. The issues with T cells are non-problematic, and they work just fine. Okay, so our next one that we're going to talk about is another B cell example. This is an IgA deficiency. This is actually the most common type of deficiency that we see. But it's not as severe as Bruton disease. Why would it not be as severe as Bruton disease? Any ideas? Yeah, you're still mature. You're still making IgM, IgG, and IgE. You just don't make IgA. So these patients do have recurrent infections that take place, right? These recurrent infections tend to be where IgA is usually found. Where is it usually found? Well, IgA is usually found within the blood and on body secretions. Okay, so they might have some infections with wounds, and they may have bloodborne infections that are circulating around. Most common type of primary immunodeficiency. Now, T cell examples. So the T cell example, we got to back up to the upper right part of the chart. So I'm going to back up real quick to the big chart. We're going to be over here. Does that make sense? So I'm zooming into right here, and that's right here. And this one's called DeGeorge syndrome. Now, in the DeGeorge syndrome, there's a partial or a complete lack of development of the thymus. Well, what's the thymus important for? T cell what? Maturation. So now you're going to have only immature T cells. You're not going to get functional T cells that are produced in DeGeorge syndrome. And you're going to get many problems associated with viral, fungal, and many protozoan types of infections in these patients. Some patients we've transplanted thymus, and it's actually worked quite well at the level of the children, and that's been pretty helpful. But again, this is just a T cell. B cell capabilities of these patients, the DeGeorge syndrome patients, is completely normal. Okay? The last one that we're going to specifically talk about with respect to primary is our combo. This is called severe combined immunodeficient or individual, or what we call SCID. In the SCID, otherwise known as an ADA deficiency, also referred to as Bubble Boy. This was David Vetter. That was the name of the boy. He was very famous for living in a sterile environment. And the SCID immunodeficiency means that there are no functional B or T lymphocytes. And so they're extremely at risk of developing all kinds of different infections. Right? They're very susceptible to bacterial infections, but also virus and fungal and protozoan as well. But again, they still have the ability to harbor a traditional inflammatory response. So if you think back to our 3 lines of defense in our introductory part of this segment, which was last unit, we talked about barriers, right? They still have mucus barriers, they still have hair, they still have skin. They still have inflammation. What they're lacking is B and T lymphocytes, which is that specialized, right? Cellular and humoral, cell-mediated and humoral-mediated or antibody-mediated response. There is a model that we use a lot that's a SCID model, laboratory model. There's a mouse model of SCID organisms. And the reason that SCID mice are used quite a bit in research purposes is you can do a lot of transplantation. You can take human cells and put them into a SCID mouse and the SCID mouse won't reject the human donation. Does that make sense? So the SCID organism is used in research purposes for a lot of stem cell type of research. So we use the SCID mouse model in my lab quite a bit because we play a lot with human stem cells. And I've tried so hard to get my students in my classroom to volunteer for some of these research experiments in my lab. But nobody seems to want to subject themselves to large full-thickness spoons on their back with stem cell transplants. Okay? But the SCID organism does a really good job of allowing for a very specific type of model to ask a question. And if you look back up, we're way up high, and you don't develop any pro-Bs or pro-T cells. Okay, so where are we on that graph? You get dizzy. We're way up here with this ADA deficiency, right? This ADA deficiency, which is the adenosine deaminase deficiency, and that leads to SCID situation, where you get no B or T lymphocytes. Okay, so primary, these are primary. The last part of today, in the remaining time, are secondary, and we're only going to talk about one example, and I think you know which one it is. It's HIV. Okay? But causes. The causes of secondary, remember, the point of secondary immunodeficiency disorder is one where it is come on not as a result of the genome. It's not congenital. So malnutrition, stress, cancer, anything that suppresses the immune system is considered a secondary immune deficiency. Okay? Bone marrow suppression as well. Even certain types of infections. You know, certain viruses like mono. Certain types of illnesses can cause a secondary immunodeficiency. You with me? But the one that's very popularized is acquired immunodeficiency syndrome, AIDS. And AIDS is the result of a human immunodeficiency virus infection. And so we're going to talk a little bit about this in the remaining time. This is a picture of the virus. A picture of the virus in the lower left is being shown in red, right? And it's a helper T cell in blue that it's actually trying to get into. And so what you see here on the right is this cartoon diagram showing the different types of characteristics of HIV. Right? It is a RNA disease, or excuse me, virus. And this RNA virus is wrapped in this sort of multiple layer from the central core. And the whole idea is to inject this DNA into the host. And it does this using some very clever mechanisms. So some of the transmission and details. HIV probably arose from simian immunodeficiency virus in the 1930s, is what our data tells us. And it began diversifying. It was first discovered in the human population in 1959 in the Democratic Republic of Congo. Transmission. Today, heterosexual activity is the #1 cause or form of transfer of the HIV virus. Heterosexual. So man to woman or woman to man. The transmission can be blood, transfusions, could be IV drug use, and we saw that very common in the 80s in this country. Sexual intercourse, again, heterosexual intercourse is the most common transmission today, but obviously homosexual intercourse or interaction also is a mode of transmission. And then the last one is mother or maternal fetal transfer. Okay, so that example that I was talking about earlier, where even if the infant or the fetus doesn't have a congenital deficiency, but mom has HIV and in so passing it on to the fetus, this is now a secondary way of getting it. There are two different strains. Excuse me. There's It's a member of the lentilvirus family. HIV-1 is more common. And it's found in patients isolated in the US, Europe, and Central Africa. HIV-2 is more common in West Africa. There's the lipid envelope that you see is actually from the host. Okay? You can see these different parts on this diagram, like the caspid protein, that P24. This is actually the one that we screen for, this P24 caspid. This is what we screen for if you have an HIV test. We're looking for that particular protein. It has a super high mutation rate. I think everybody knows this now. It has a higher mutation rate even than the flu virus. And the data that I actually want to talk about is this pathogenesis of HIV. It targets the immune system, and the way that it gets in is actually quite clever. So here's our HIV virus, and it has these docking proteins. And you can see this GP120 docking protein finds our T helper cell, right, CD4. And as it docks, it goes through a conformational change. And this conformational change allows for the GP120 to bind more aggressively, and allows for this GP41, which is found right here, found within the GP120. But when it goes through this docking, now this is what burrows and penetrates into the membrane, and then it injects this P24 wrapped caspid. Okay? Well, what facilitates this docking is this receptor. This receptor that's actually found on CD4 cells. So this receptor is the CCR5 receptor. And there's actually a patient population that doesn't have this receptor. And we discovered that these patients are immune to HIV. They cannot be infected with HIV because they lack this CCR5 receptor. Okay? This is about as much as we know now, as much as I know. And I'm by no means an expert in HIV, but want to bet that a lot of area of research in the future that you all will be hearing about is probably something to do with this docking. You can prevent the docking. You can have the virus in you, but it never affects you because it never actually is able to penetrate. Okay? So without CCR5, you actually are immune to HIV. Now, what happens when a typical patient becomes infected? Well, there's an acute infection that takes place, and you can see here is up top a CD4 positive T cell, a helper T cell. Both helper T cells and dendritic cells can both be infected with the virus. Both dendritic cells and helper T cells can both be infected with the virus. They both have the CCR5 receptor. Okay? So they get infected with the virus. They make it into the lymph node, and you start developing anti-HIV antibodies, and this spreads throughout, this is part of this viremia phase, and this spreads throughout the body, and then you get into what we call this latency phase, where the virus stays a little dormant in the infected cells. And then later on, when it decides that it's time, and it's an opportunistic virus, there could be a stimulator, a stressor, or a stimulator that takes it out of its clinical latency, and once it is out of its clinical latency, it moves into full-blown AIDS, which is acquired immunodeficiency syndrome. And really what's happening in this phase is what's going on in this slide. So we've got our primary infection, and this is the way you read. This is kind of a difficult slide to read, so this is our last one. Let's just walk through it though. CD4 positive T cell populations. You can see there's about 1,000 cells per cubic millimeter. Okay? Here's our primary infection, and the number of surviving or resident healthy CD4 positive T cells, helper T cells, drops down. Then you get this clinical latency where it stays sort of plateaued. And then at some point, it drops significantly. These are years on the bottom. This little hash mark tells you that it's not necessarily shown to scale. But this clinical latency period is when patients have HIV, but they don't have symptoms of AIDS. And when this starts to drop is when you see, here is our primary infection, you get a massive increase in the HIV RNA copies, right? And then it kind of drops down, and it stays somewhat plateaued during clinical latency. And then at some point, it starts to shoot up. And when it shoots up, it kills off the rest of the CD4 population. This is where you have AIDS, okay? And the AIDS really is death due to opportunistic diseases. I mean, AIDS patients die from pneumonia just as much as they die from other types of infections, because their helper T-cell population is so low that they don't have a functioning immune system anymore. And so like a common cold to an AIDS patient can kill them. Okay? And that's what's going on with a secondary immunodeficiency disease, our example being HIV. Questions? All right, remember post-quiz Friday, because last week we were out of the routine.